हाय गाइस, वेलकम टू आवर यूट्यूब चैनल नीट भाई का सिंपल ज्ञान। तो आज हम लोग शुरू करने वाले हैं हमारा क्लास 11 का बायो और बोटैनी का चैप्टर बायोलॉजिकल क्लासिफिकेशन। सो रैपिड रिवीजन करेंगे। 10-15 मिनट में इस चैप्टर को निपटा देंगे, ठीक है गाइस। तो आज हम लोग शुरू करने वाले हैं बायोलॉजिकल क्लासिफिकेशन। तो लेट अस स्टार्ट। First scientific classification done by Aristotle divided plants into half shrub trees, animals into anima and anima. Anima no red blood cell, and anima with red blood cell. So two kingdom classification given by Linnaeus, kingdom Plantae and Animalia basis, cell wall, locomotion, mode of nutrition, response to stimulus and contractile system. Three kingdom classification given by Ernest Haeckel introduced kingdom Protista. Four kingdom classification given by Copeland introduced kingdom Monera. Five kingdom classification given by R.H. Whitaker introduced kingdom Fungi. Based on characteristics of five kingdom classification, kingdom Monera, Protista, Fungi, Plantae, Animalia. Kingdom Monera, unicellular, prokaryotic, autotrophic and heterotrophic. Kingdom Protista, unicellular, eukaryotic. Unicellular eukaryotic is kingdom Protista. Monera is unicellular prokaryotic. Protista, autotrophic and heterotrophic. Fungi, Plantae and Animalia are multicellular eukaryotic. Fungi is heterotrophic, Animalia is heterotrophic, Plantae is autotrophic. Based on cell organization, thallus organization, mode of nutrition and reproduction, phylogenetic relationship. Note: Chlamydomonas, chlorella, Euglena, amoeba, paramecium were previously classified differently based on cell wall, but now brought together under kingdom Protista in five kingdom classification. Chlamydomonas, chlorella, Euglena have cell wall. Amoeba, paramecium. Chlamydomonas, chlorella have cell wall. Euglena, amoeba, paramecium does not have cell wall. Previously under different system of classification based on five kingdom classification brought under kingdom Protista. Sixth kingdom classification given by Carl Woese based on 16 srRNA sequence gene coding divided into three domains: Archaebacteria, Archaeubacteria, eukarya, originating from a common ancestor progenote. Archaebacteria, thermostophiles, halophiles are methanogens. Eubacteria or true bacteria, eukarya, kingdom Protista, fungi, Animalia, Plantae. Kingdom Monera, bacteria. Sole members are unicellular prokaryotic bacteria. Reproduction mainly by binary fission. Divided into Archaebacteria and Eubacteria. Metabolic diversity in Monera: obligate anaerobe Bacillus subtilis, obligate anaerobe Clostridium, facultative anaerobe Pseudomonas, phototrophic oxygenic cyanobacteria, anoxygenic purple sulfur, green sulfur bacteria. Chemoautotrophic or nitrifying bacteria: Nitrosomonas, Nitrocystis, Nitrobacter. Type of bacteria: Archaebacteria. Living fossils: extremophiles, methanogens, halophiles, thermoacidophiles. Methanogens: Marciarias, example Methanococcus methanobacterium, obligate anaerobe chemoautotroph. Halophiles: Saltarias, Halococcus halobacterium, facultative anaerobe heterotrophs. Thermoacidophiles: hot spring, thermoplasma, thermoprotease, facultative anaerobe chemoautotroph. Cell wall structures: pseudomurine contains NAG-NAT instead of NAM. NAT instead of NAM. Plasma membrane: phytolin branch side chain with lipid monolayer provides heat resistance. Genetics: has a distinct gene that codes for histone protein. Cyanobacteria, blue-green algae, also called blue-green algae, cyanobacteria, contains chlorophyll A. Phycobilin pigments, can be unicellular, colloidal, filamentous, freshwater, marine or terrestrial. Possesses specialized nitrogen fixing cells called heterocyst. Example Anavina, nostoc, oscillataria. Forms trichome. Forms blooms in polluted water bodies. Mycoplasma, true bacteria, cyanobacteria and mycoplasma are true bacteria. Archaebacteria are classified as different. Mycoplasma, also known as pleuropneumonia-like organism PPLO or MLO molecule-like organism known as joker of plant kingdom due to pleomorphic nature. Smallest living cell 0.1 micrometer to 0.3 micrometer, completely lacks cell wall, therefore resistant to penicillin. Bacteria with their coats of plasma membrane has a trilamellar structure, double stranded linear DNA, facultative anaerobe, highly resistant to penicillin as penicillin attacks cell wall, cause disease like little leaf in brinjal. Mycoplasma affects mostly plant but can affect both plant and animal. Key bacterial disease chlorella caused by Vibrio cholera, typhoid caused by Salmonella typhi, tetanus caused by Clostridium tetani, citrus canker caused by Xanthomonas citri. Now, kingdom Protista given by Ernest Haeckel. So we have done till kingdom Monera. Now we'll go to kingdom Protista, next kingdom, then kingdom fungi, then Plantae and Animalia. Kingdom Protista, unicellular eukaryotics, primarily aquatic, connecting link between Monera and other kingdom. In kingdom Protista, boundaries are not well defined. Shows both sexual and asexual reproduction. Favorable conditions for divide by binary fission. Unfavorable condition cyst formation. Number 1 kingdom Protista, chrysophytes, then dinoflagellates, then euglenoids, slime molds, and protozoan. Chrysophytes, phytoplankton or golden algae. Includes diatom and desmids. Diatom, desmid also known as golden algae. Microscopic and float passively in water current, mostly photosynthetic. Cell wall is embedded with silica, thus indestructible. Forms overlapping cell-like soap box-like. Upper lid epitheca, lower lid hypotheca, in between protoplast. Reserve food Chrysolaminarium. Diatoms are the chief producer of ocean. Accumulation of cell wall forms diatomaceous earth, Kesselgur. Excellent pollution indicator disappear in polluted water bodies. Dinoflagellates, mostly marine, photosynthetic. Cells have rigid cellulosic plates, armored dinoflagellates. Biflagellated, produce characteristic wheeling whips motion. Contains chlorophyll A, chlorophyll C, haplontic life cycle. Bioluminescence exhibited by Noctiluca. Red tide, rapid multiplication of gonlox and gymnodium colors the seed red. Produces toxin causing paralytic shellfish poisoning PSP via gonlox catilina. Euglenoids, mixotroph, freshwater found in stagnant water bodies, plant-like, animal-like, photosynthetic in light, heterotrophic in dark. Therefore, plant-like and animal-like. Biflagellated, one short and one long flagella. No cell wall pellicle present, proteinaceous pellicle. Has myonema for characteristic gliding movement. Reserve food paramylon. Examples Euglena, Paranema. Slime molds, protistian fungi, saprophytic consumer decomposer, heterotrophic. Under favorable condition, aggregation forms plasmodium. Under unfavorable condition, forms footing bodies bearing spores. Spores have true cellulosic cell wall, highly resistant. Types acellular, cellular, acellular diploid, cellular haploid. Acellular diplontic life cycle example Physarium, Physaria, Fuligo. Cellular haplontic life cycle example Dictyostelium, Polisondium. Protozoan, primitive ancestor of animal kingdom. All are heterotrophics, cell wall absent, covered by pellicle. Four major groups are amoeboid protozoan, flagellated protozoan, ciliated protozoan, sporozoans. Amoeboid protozoans via pseudopodia. Marine form have silica cells. Example amoeba, antiamoeba. Flagellated protozoan have flagella. Example trypanosoma, cause sleeping sickness disorder. Ciliated protozoan aquatic, actively moving using thousands of cilia, gullet opening outside. Example paramecium, sleeper organism. Sporozoan have infectious spores like stage, locomotory organs absent. Example plasmodial, malarial parasite. Now kingdom fungi. Multicellular eukaryotes Given by R.H. Whitaker 5 kingdom classification, dominant phase is gametophyte haploid. Heterotrophic absorbers, cosmopolitan distribution, great diversity in morphology and habitat, cell wall composed of chitin and polysaccharide, reserve food glycogen, specialized habitat, growing on dung known as caprophilus, hair or nails keratinophilus. Vegetative structure consists of thin filament called hyphae. Network is called mycelium. Accepted and synocytic phycomycetes, septate and branched ascomycetes, basidiomycetes, and deuteromycetes. Reproduction steps: plasmogamy, then karyogamy, then meiosis. Asexual spores are conidia, sporangiospores, and zoospores. Sexual spores are ascospores, basidiospores, and oospores. Phycomycetes, lower fungi, habitat aquatic decaying wood, obligate parasite on plant. Mycelium accepted synocytic. Asexual reproduction by zoospore or aplanospore non-motile, produced endogenously. Sexual reproduction zygospore, formed by fusion of gametes, divided into oomycetes, zygomycetes. Oomycetes example: Pythium albugo, Saprolagnia phytophthora. Albugo, white rust of crucifer. Phytophthora, late blight of potato. Zygomycetes, conjugation fungi example: Rhizobus, bread mold, mucor mold. Ascomycetes, sac fungi. Habitat: saprophytic decomposer, coprophilous or parasitic, mostly mesocellular except yeast unicellular, and Saccharomyces. Mycelium, branched and septate. Asexual spore conidia, produced exogenously. Sexual spore ascospore, reproduced endogenously in sac-like structure called asci, arranged fruiting bodies called ascoscarp. Prominent dikaryophase. Example: Neurospora, Drosophila of plant kingdom, used extensively in genetic word. Penicillium, Aspergillus, claviceps. Edible forms are morels and truffles. Basidiomycetes, club fungi. Habitat: soil, log, tree stump, parasites, rust and smut fungi. Mycelium is branch septate, long dikaryophase. Long dikaryophase more than Ascomycetes. Asexual spores are generally not found. Vegetative reproduction by fragmentation is common. Sex organs are absent, but plasmogamy occurs by fusion of somatic cells. Sexual reproduction: basidiospores are produced exogenously, basidium following karyogamy, and meiosis. Fruiting body called basidiocarp. Example: rust fungi, Puccinia, smut fungi, Ustilago. Edible: Agaricus, poisonous, Stadstule aminata, Ganoderma, bracket fungi. Last fungi, imperfect fungi, Deuteromycetes. Imperfect fungi, sexual stage is absent. Mycelium, septate branch, cheaply decomposer, help in mineral cycling. Perfect sexual stage are discovered, they are moved to Ascomycetes or Basidiomycetes. Example: Gibberella fusicori. Asexual reproduction: exogenous conidia. Example: Fusarium alternaria, Coltotrichum, Trichoderma. Last, lichens and acellular entities. Virus. Lichen, symbiotic association with algae, Phycobian 5%, mostly blue-green algae. Fungi, Mycobian 95%, mostly Ascomycetes. Highly sensitive to sulfur dioxide, SO2. Excellent pollution indicators, do not grow in polluted areas. Corrode rock to initiate soil formation, used in foods, dyes, perfumes. Virus, acellular structure, historic milestone: Ivanovsky discovered tobacco mosaic virus. Bejernik coined the term contagium vivum fluidum. Stanley, crystallized virus. Genetic material, either DNA or RNA, is present in virus. Double stranded DNA in T4 bacteriophage. Single stranded DNA in M13 phage. Double stranded RNA in reovirus. Single stranded RNA in TMV virus, retrovirus, influenza. TMV and T4 bacteriophage is important, given in NCERT. Viroids discovered by Theodoner, smaller than virus, consist of free low molecular weight RNA without protein code. RNA without protein code is present in viroids. Infects plants only. Cause potato spindle tuber disease. Prion, infectious proteinaceous particles, genetic material is absent. Abnormally folded proteins resistant to heat and enzyme. prions are same size with virus, causes neurodegenerative disease, Creutzfeldt-Jakob disease, Bouin spongiferum encephalopathy, scapie in sheep, Kuru disease in humans. That it's for biological classification with 15-minute rapid revision. Thank you.